Awesome. Uh, so thank you, everyone. So this is Tausur Rahman. I'm a PhD candidate in biomedical engineering at Johns Hopkins University. I'm working with Ramachalappa, Professor Ramachalappa. So today I'll be talking about scalable and precise histopathology, especially the next generation of deep learning methods that are helping us make computational pathology faster, smarter, and more reliable. So over the next few minutes, I'll walk through two of my research contribution that have come out from my PhD. One is published in Nature Modern Pathology, and one is recently published in MIDL, Medical Imaging for Deep Learning, which addresses two biggest challenge, especially in whole slide image analysis in digital histopathology, uh, like representation quality and computational efficiency. Uh, Before we dive in, let me give you some overview about the presentation. We'll start with the context of what histopathology actually is and why whole slide images create a unique computational challenge in medical AI. And then we'll look at the problem, the generalization gaps, and computational cost that make current AI approach a little bit challenging. From there, I'll present our two solutions. The first is the framework called TS-SSL, which is Task-Specific Self-Supervised Learning for computational pathology. And then, if I have time, I'll talk about dynamic token compression, which used for reducing the computational cost. Uh, let's start with the basics. What exactly is the histopathology? Uh, At its core, histopathology is the microscopic examination for the tissue to study the disease, especially in the cancer, or like it's a gold standard to diagnose different cancer subtypes, also the grading. So the workflow has like three standard steps. Like first, a biopsy. So whenever there's like any sort of symptoms, they do biopsy, which a small sample of tissue is taken from the body, usually under the anesthesia. And secondly, they do the staining, which is the tissue is treated with the H&E, hematoxylin and eosin dyes, which turn different cell structure into pink and purple color, then making them visible under the microscope. And finally, diagnosis. A trained pathologist examines the stain slide. Um, and they look for the abnormalities, um, like how cells are arranged, like their shape, um, their size, or their structure of the abnormal cell. And what's important to understand that just how expert intensive this process is takes year of experience for the pathologist to find out uh, this abnormality in the tissue. And it's also not, it's very variable. It's like a different pathologist has different opinions on the same slides or same images. So that's exactly where I think AI can add value to assist the pathologist to scale and make the process more consistent. Now here's where things get interesting for a computational standpoint. Today, like those glass slides, whole slide images are being scanned into digital images. Uh, and these are not like usual medical images. A single whole slide image can be 45,000 by 30,000 pixels on average. It might be more than that as well. To put that in perspective, that's equivalent to stitching like around more than 500 iPhone images. So it's a computationally huge image. So when a pathologist uses a microscope, they are looking at the small field of view at a time. They manually navigate and try to find out like the the different abnormalities of the cells. Uh, so cancer cell, another problem is like for the whole slide image, the cancer's signal to noise ratio is very less. Like suppose like it's less than 3% of the full image has the cancer area. So it's a very challenging task to figure it out like, where is the actually the cancerous tissue. Uh, Here's the comparison between the traditional histopathology and digital histopathology. In traditional histopathology, uh, uh, it's prepare the slide scan and then it examine under the physical microscope, and then they store the glass in the hospital for a decade, and it's very manual, and sometimes the quality of the slide degraded, so it's tough to retrieve after a long time when they do the uh, analysis. But in digital histopathology, the same stained uh, slide is scanned using the whole slide image scanner. Now that image lives in the cloud or a server, and it can be shared instantly with the multiple expert. Then multiple expert can easily give their opinion on it. It also can be analyzed by the AI algorithm, and it is the quality degrade uh, in cloud. Uh, s- as I said, like the it's computationally very expensive because of the gigapixel image, and there are like very common deep learning workflow. Uh, is like they don't directly put the full image to the deep learning model. They there are like some different steps, like whole slide image. They firstly try to segment the tissue uh, from the background, and then uh, what we do is like extract the patches from this tissue uh, and use these patches. So one whole slide image has thousands of patches, like it might be 5,000 to 10,000 patches, and then we use these patches to the uh, downstream task for the classification or segmentation or any other downstream task. So there is few challenge. It's like uh, it's still because one whole slide image, if it has thousands of patches, so if we have the sample sizes of like suppose few thousands of patients we have, then it's millions of patches are there. So it's very tough to directly train the model in the clinical settings. And what the usual approach is like extract the features from the each patches, and then we use these feature embeddings for the downstream task. Um, so for this challenge to recover, uh, we propose our solution like task-specific self-supervised learning, which uh, which also compared with like different foundation models and different regular approach, which as a baseline. If I say some background study, like mm, in computational pathology, because of its gigapixel images, uh, for when they started AI in this domain, they used to use some ImageNet pre-trained encoders to extract the features from the patches, because it's like, I mean, like ResNet 50 or some other CNN pre-trained network. And last few years, there are some foundation model uh, developed by the researchers, and they start using the foundation model. which pre-trained on the pathology images. The issue is like there is trained on like thousands of different cancer images. Uh, so it's make more generalized. Uh, and there are, if you see the figure here, it's like there are different cancer data set like lymph node or colorectal. So the patches are very different from each other's. So whenever we use the foundation model, it extract the features which is uh, more high level, and we miss some sort of variability when it comes to different task-specific feature extraction. So our idea was like if we can train a model which only pre-trained on a specific task, like if we want to do some downstream tasks for breast cancer or lung cancer, we'll just train the model some subsample of breast cancer data and then see how it works for this downstream type for this task a specific way. And so if you see this, like this, our idea is like we have the patches and we take subsample of the patches from the data set, and then we train an encoder in a self-supervised manner or for the same task, and then we use this encoder for the downstream task for this same sort of uh, um, data. Like if like it's breast cancer, then it's the subsample of the data would be the from the breast cancer. The nice thing is like, I mean, we saw is like some improvement whenever we see the foundation model and the task-specific things. Uh, so this is our approach. If you see the framework, there are like two different stage. One is like a self-supervised manner, and then one is supervised manner. So we have the tiles dataset here, and we train a models in self-supervised manner where we use like subsample of the main full dataset, and then we train a autoencoder like a special attention and channel attention based. So and then after training this self-supervised approach, we use this encoder as a frozen encoder for the downstream task. And the final part is like the classification task, like where we use attention-based MIL because of mostly whole slide images. We have a slide level, and a special attention block is a very shallow block where we are trying to find the special information from the patches, and then also we use the channel attention to get the channel information, and we use the different, I mean, regular reconstruction laws and the cross entropy laws for the classification uh, task. Here's the performance of whole slide classification task. If you see, we use different encoder or so so there are like we did two different classification tasks. One is like whole slide classification task where we have whole slide slide level. That means we have level for the full slide, and we also use some sort of pass level dataset where we have level for the pass. Yes. And if you see the table here, it's like we have different encoder, and some of the encoder which is like CNN encoder, uh, we use the random initialized or ImageNet to see the performance difference, and also use the pathology foundation model based encoder, ResNet FM, KimiaNet, and Flip, which trained on large number of pathology image dataset, and we compared with different benchmark dataset. So we did three different cancer data, NSCLC, which is based on lung cancer, and also BRCA is breast cancer, and PANDA is prostate cancer. And what we see, if you see the performance comparison, um, The performance between like ImageNet pre-trained CNN model and the TSSL task-specific model has like almost 4 to 5% improvement here. And if you see the foundation model, there are like a few percentage of improvement we can see over all the data set. So which shows that there are like some sort of gap uh, task-specific self-supervised learning is filling from the foundation models feature extraction. If you see the best classification task, it's also showing the similar pattern of we have Panda, CRC, and PCAM, which are so Panda is like the same data set what we use for the classification task, but our pathologists, they also leveled them as a patch level. So we also can use them as a patch classification task. And there is CRC, which is uh, kidney cancer dataset. And also we have PCAM, which is node metastatic dataset, where we try to see is like the cancer is the binary class, like normal and abnormal. And we see the same pattern, like if the our idea is like to use the task-specific encoder for different tasks is performing better rather than the generic foundation model, which train on huge data set. And yeah, and also it's better to don't use the ImageNet based P10 encoder for this sort of medical image, histopathology images. And we also try to see the visualization, like actually how the feature extraction actually working here. So this Disney plot we got from for the CRC dataset, and we have 4 different plots. If you see the first one is random initialized using the ResNet 50, and this one is like ResNet 50 ImageNet pre-trained, and the third one is pathology pre-trained network, which is called FLIP, which is the based on the CLIF model. So they trained for the using the uh, pathology images. And the last one is our approach. So we also calculated some score to get the quantitative difference, like Shulity coefficient and Davis-Baldwin index, which also showing that like if the Shulity coefficient is high, that means the classes has more clustered separate cluster, and then Davis-Baldwin index is lower, means is showing that uh, it's actually better clustered. So if you see the visual and then quantitative both the approach, our approach showing little bit better improvement than the foundation model in Flip, and also the if you see the ImageNet based and random initial based is like more the cluster is not very separated here. So this like this I mean these are uh, conclusion that like we tried different approaches for to encode the features because it's very important for whole slide images. We can't directly use the end-to-end training for whole slide images in computational pathology. So which types of feature encoder we are using is actually very important. And mostly people use the foundation model, but our idea is showing that if we can do some pre-trained on a specific task-specific data set, it performed better for this domain. Yeah, I think I can conclude my presentation here. If you guys have any uh, question or any comments, feel free to answer.